on such a cold spring day in Melbourne. On behalf of the Australasian Society for Stem Cell Research and the National Stem Cell Foundation of Australia, I'd like to welcome you to our forum tonight, Gene Therapies and Stem Cell Research, Now and in the Future. My name is Megan Munsey, and I'm a researcher at the Murdoch Children's Research Institute and at the University of Melbourne. I'm also the Vice President of the Australasian Society for Stem Cell Research and a Director of the Foundation. And tonight, I'm delighted to be your host. I'd like to begin by acknowledging the Wurundjeri people, who are the traditional custodians of the land on which we meet tonight. And I'd also like to pay my respects to elders, both past and present, of the Kulin Nation, and extend that respect to other Indigenous Australians who are joining us at the State Library or listening online to the recording. Tonight, we're here to discuss what is gene therapy and how Australian researchers are trying to develop new treatments using this technology. In a nutshell, gene therapy involves altering the genes inside your body's cells in an effort to treat or to stop disease. Genes contain your DNA, the code that controls how our body develops and functions. Genes that don't work properly can cause disease, and gene therapy holds the promise for treating a wide range of diseases, including cancer, cystic fibrosis, blindness, haemophilia, and even AIDS. Last month, Associate Professor Lauren Ayton and colleagues published findings from a study where they asked Australians with inherited diseases that affect vision whether they'd be likely to accept gene therapy if it was available to them or to their families. Over 90% indicated that they would, but only about a third agreed that they had a good understanding of what this would involve. Tonight, I hope we can provide a little bit more clarity on how, what, why, and when gene therapy can be used now and in the future. To help us in this quest, we're going to hear from 6 fantastic speakers tonight, all leaders in their field. I'll introduce each of them in turn, but they're going to tell you about what they find interesting about gene therapy. And at the end of hearing from each of our speakers, it's going to be over to you. We're going to have an open mic section where you can ask the panellists questions that you're dying to know. And it doesn't matter what that is. I think we're keen to hear from you, and we really want this to be an open exchange between these experts, but also making sure that you're included in the science. So our first speaker is Dr. Ashley Young. Ashley is a haematologist based at the Peter Mac and the Royal Melbourne Hospital. He has expertise in treating patients with acute leukaemia and other diseases affecting the blood system. He's also a researcher at the Walter and Eliza Hall Institute for Medical Research, and he's looking into how blood cells are produced from stem cells and what goes wrong in blood disease. So Ashley, over to you.
Thank you so much, Megan, and lovely to see so many faces on a Melbourne evening. So that's a nice euphemistic way of placing it. Yes, I'm Dr. Ashley Young. I'm a haematologist, as Megan told you. I work at my research from the Walter Eliza Hall Institute of Medical Research, but I also work as a clinical haematologist, mainly treating patients with acute leukaemia at the Royal Melbourne Hospital and Peter MacCallum Cancer Centre, so all fairly close by within the precinct. Oops, I think I broke it. Didn't take me long. So what do I do? So I'm a clinician researcher, and that means I research in terms of trying to understand the diseases that I treat, but also I treat patients as well. And my research is really focused on looking at factors which help regulate blood cell development and stem cell development, and create, to make a stem cell, which is a very basic blood stem cell, into the cells in your body. So blood stem cells were one of the first stem cells ever characterised in history. They were known to have specific properties in terms of being able to recreate the entire blood system of an organism in humans. They're responsible for making eventually the red cells, which carry oxygen in the blood, the white cells, which fight infection, and platelets to actually stop people bleeding. And these are the fundamental blood products which actually keep us healthy. But the way that blood stem cells do that is actually fairly complicated, and how we actually understand a blood stem cell becoming those cells is one of my areas of research. I'm also very interested in how abnormalities in the genes that control these processes can also lead to blood cancers. So stem cells have been actually used by us as haematologists for decades. And the reason that we're able to give patients chemotherapy for treating, for example, their leukemia, is the ability of these stem cells to resist death from giving our chemotherapy agents. When we give our treatments to our patients, we have them in hospital for a good period of time because the chemotherapy agents cause a fall in the numbers of the white cells, the red cells and the platelets that I told you about before. These resistant cells are able to repopulate the entire patient's blood system, but happens over a period of weeks. So these unique properties of stem cells we actually use in terms of our treatment every day. We've also been able to use our stem cell treatments as living therapy. So for some patients with certain diseases, we give such high doses of treatment with chemotherapy and or radiotherapy that without giving the patient's own stem cells back to them, they would have significant complications of bleeding and infection. And so that's what we call autologous stem cell transplantation. And the contribution that we've made in Australia was from this gentleman named Don Metcalf, who identified factors which actually could stimulate the stem cells being released from the bone marrow and into the blood. And the way that that's what he used, something called GCSF, or granulocyte colony stimulating factor, to give to stem cell donors. So, for example, in the left-hand side, you can see our traditional way of sucking out the bone marrow from patients to actually access the stem cells. We have now, predominantly in adults, used that machine to actually able to collect the stem cells from patients after administering the GCSF therapy. So that's been a fundamental scientific breakthrough led by Don Metcalf, which has actually changed the way we've collected stem cells across the world, with over hundreds of thousands of donors being collected this way now. One of the important things, however, is to actually think about using stem cells from someone else. So for certain diseases such as acute leukemia, we actually know that giving them chemotherapy, the chemotherapy unfortunately fails them. And when we've given autologous stem cells and higher dose chemotherapy, that doesn't really impact on how their survival and how we can cure them. What has been found out is that if we can sometimes give the stem cells from someone else to the patient, we can actually cure them of leukemia, including leukemia which has been resistant to treatment from chemotherapy. And the reason for this is that ability of those stem cells from someone else to reconstitute the immune system. But it's not the immune system of the patient, but it's the immune system of the donor. And that donor immune system, which recognises the leukemia as foreign, is able to actually destroy those the leukemia cells and recognises them not natural to it, and that was what results in the cure. So those are important things, because one of the two things when we're talking about from the next couple of things is that using those technologies over the last few decades, we've been able to now to use, for example, autologous stem cells, that means the patient's own stem cells, if they had a defect, and correct some of those with gene therapy. So for patients with certain blood mutations, such as thalassemia or sickle cell anemia, where there's a mutation in the red cell gene which causes the red cells to behave abnormality, if that abnormality can be corrected or mitigated by correcting the genes in the patient's own stem cells and then giving those stem cells back to the patient, we can actually reduce the severity of that disease. And certainly that's been shown in the last few years that can be an effective therapy for diseases which would otherwise not be curable. Similarly, if we can actually use the immune cells rather than the stem cells by manipulating the genetics of the immune cells to use those cells to attack the leukemias, that means we can use these as living therapies against leukemia. So for some of these therapies you may have heard of are what called CAR T cells, and those mean chimeric antigen receptor T cells, which are like directly targeted T cells from a patient to the leukemia of the patient. So what I'm talking about here is looking at the last few decades where we've used our understanding of stem cells and married that with our ability to understand the genetics of diseases, but also manipulate those genes in the stem cells to actually be able to help cure patients of other diseases, but also to use that same technology that we've learnt about to manipulate other cells of the blood system to use to treat leukemias which has otherwise been unable to be cured with chemotherapy or bone marrow transplant. So I'll leave it there, and thank you.
Thanks so much, Ash. So our next speaker is Dr. Sandi Kariyawasam. Sandi is a paediatric neurologist from the Sydney Children's Hospital in Randwick. She's also a lecturer at the University of New South Wales, and Sandi's doing a PhD there too. And as part of a PhD, she's following the progress of children who've been treated with novel therapies who have muscular and nerve problems. Sandi, over to you to tell us about your interests and role in gene therapies.
Thank you, Megan. I've actually finished my PhD. Thank you, everybody. So I'm going to take a slightly different tack to Ash. I'm also a clinical researcher, so my interest is taking exciting scientific breakthroughs and embedding them into a clinical pipeline, so really getting from bench to bedside. I've lost the... Do you have the control? I can't see it. Oh, it's here, sorry. Apologies. Oh, beautiful. Okay, so I'm going to give a brief overview of the triumphs, trials, and some tribulations that we have gone through at Sydney Children's Hospital Network and UNSW in getting an SMA gene therapy pipeline. SMA is spinal muscular atrophy. So, to me, a picture speaks 1,000 words. Many of you will know the story of Mackenzie Casella, who was born 6 years ago in 2016. As a clinician, my heart used to sink when I saw babies like Mackenzie, who were weak and floppy and had a diagnosis of spinal muscular atrophy, or SMA, because in 2016, this was an untreatable disease, the leading genetic cause of infant mortality in the West, where irreversible loss of motor nerves would lead to muscle wasting and weakness. Over time, a baby's ability to breathe, feed, and move would deteriorate. Mackenzie, like 95% of children just like her, would not see her second birthday. In 2 short years, though, we had hope. A new frontier of advanced therapeutics, including gene therapy for the condition, showed the potential in clinical trials to change the course of SMA, to modify the way the disease progressed, but these therapies were not a cure. Aviana, shown here with her family, unlike Mackenzie, did survive and made improvements in motor function with treatment. But although this was a success story to many and was in the newspapers as such, her parents felt devastated that she couldn't be treated earlier. Because she was treated after her disease had progressed, she was left with motor deficits and difficulties with function, including wheelchair dependency. Our SMA families kept echoing this truth. In rare diseases, a lack of awareness leads to a struggle for an efficient and accurate diagnosis, limitations in access to therapies, and support through the healthcare journey. We've heard this time and time again in clinic. So we recognized early on that you can't just work on one of these aspects in a silo. You've got to have simultaneous excellence in timely diagnosis, access to therapies, and within the health system. So why am I sharing these stories with you? Well, firstly, to show you how quickly the therapy landscape in rare neurological diseases can change and was changing in SMA, but also to highlight that our patients then and now set the compass for the next phase of the clinical and research journey. They were and really still are a call to action, showing us that there's still a host of unmet needs in the SMA community and giving us the impetus to ask, how can we do better every day? As the next frontier, that of gene therapy, will surely bring with it triumphs, trials, and tribulations. So how can we do better? For us, embedding a culture of research into clinical care is vital to translate science into real, meaningful gains for our families. And that's where our SMA communities, like you, and rare disease communities come in. Your voice should be at the center. For our families, they don't have the time to wait for years for access to potentially life-saving treatments, as the clinical course is degenerative and sometimes precipitously degenerative. So there's a real impetus to establish clinical trial readiness as a pathway through which families can access these emerging therapeutics. Horizon scanning is therefore really vital in this rapidly changing therapeutic landscape. On the back of gene therapy trials for symptomatic children with SMA, and gene therapy here is where you replace the missing copy of the SMA gene, SMN1, in an envelope of a virus called AAV9, and you give it through systemic infusion. So it's a one and done procedure, or it should be. So what we did was that we saw the symptomatic trials and we saw children were improving with them, and we had our finger on the pulse and we knew that pre-symptomatic trials were the next step forward. Children dosed before the disease onset potentially could do better than the children who were dosed with symptoms. So we did something actually quite novel. We, as a centre, approached the trial sponsors and showed them that Australia had the research capabilities and the infrastructure to be part of the seminal gene therapy trial for pre-symptomatic SMA, where children would be dosed before they started showing disease symptoms and before the age of 6 weeks of age. But accessing gene therapy and in a newborn population is very different from taking part in a trial for conventional medications. We essentially had to develop and implement 2 pipelines in parallel and one from scratch. Both from scratch. One where we could accurately establish pre-symptomatic diagnosis through newborn screening, alongside a gene therapy pipeline that allowed us to screen children for safety to receive the drug, establish logistics to get the drug from overseas, because Australia doesn't have the manufacturing capabilities to get the viral vector, so we had to get that in from America, and this is all through a COVID pandemic. Store and administer the gene therapy safely, and have a robust clinical follow-up pathway to monitor for safety and effectiveness. So yes, these breakthroughs are exciting, but there's so much more to getting them to the bedside than just giving it to the patient. There's lots of steps in between. And you've got to support these families. They receive potentially devastating diagnosis for their newborn in what should be a happy time. We had to counsel them through the risks, benefits, and uncertainties of treatment before they could make an informed decision to have gene therapy by the time their baby was 6 weeks of age. This put pressure on our workflow, as essentially these families and babies, they took precedence. They were regarded as a medical emergency. You cannot establish these pathways with such tight timeframes without proactively fostering a spirit of communication and collaboration across all stakeholders. We have learned that we can't afford to work in silos, and I think the greatest triumph of newborn screening for SMA and the gene therapy pipeline is that there has been such immense efforts to coordinate and work alongside institutions that are pretty used to historically working by themselves. Screening service, health, industry, patient advocates, communities, policy stakeholders, they all came to the table. And we had 8 children successfully be part of this inaugural international trial, with treatment intervention by on average 13 days of age. And this time frame was a testament to that coordinated care, making sure that no child or family fell through the gaps within that healthcare journey. But as we all know, a clinical trial is just the tip of the iceberg. and there's inherently inequities in access by its very design. So that's where our real-world studies come in, and they're aimed at overcoming these inequities and providing evidence to inform best clinical practice and change health policy. And what we did was we evaluated the accuracy, implementational pathways, public acceptability, clinical usefulness, and of course, cost analysis of newborn screening for SMA, coupled with often high-cost therapeutics. And we presented that to the Newborn Screening Standing Committee, and that was part of the evidence base for policy change. And as of 2021, SMA has been advocated for to be part of the routine screening panel, which is a major breakthrough. And lots of the states are doing their own pilots. New South Wales, ACT, have already embedded it into their newborn screening program. So I just have a few more minutes of your time and attention. As we have developed this robust translational pipeline for gene therapy, we've been able to dose 24 children. That's neonates, infants, and children who are about 6-7 years of age with gene therapy. As the access has shifted from clinical trial populations through to managed access programs, and now is part of our clinic through the PBS reimbursement scheme. We refine our pathways continually by using research as a tool to understand where we could do better, driving equity and efficiency of diagnosis and therapeutic access, whilst minimizing risks and maximizing benefits for our family. And I end with one of our triumphs. This little 13-month-old girl who was treated with gene therapy is taking her first steps in clinic. And that is amazing when you think what her trajectory would have been 6 years ago. We wouldn't be having this kind of conversation. And actually gets a bit of a lump in my throat. She's expected to have a normal developmental trajectory. I don't know if I can play that again, because it's such a nice video. Maybe not. Oh, there you are. She is a lasting legacy for all children who came before her, like Mackenzie, like Aviana, showing that there is great hope if we work together to develop these pathways for rare diseases. Thank you for your attention. I'll leave that there. Oh, sorry.
Fantastic. Thank you so much, Sandi. So our next speaker is Julie Sini. And I was actually thinking as Sandi was speaking, maybe I should say you are the voice. Because Julie's going to talk to us about her role in advocacy. So Julie is a Melbourne-based community leader who's a fierce campaigner and has been over the last 15 years advocating to provide better access to treatments for Australians living with SMA. Julie, please share your story.
Got to get the button right. Thank you everyone for your time. I don't have any letters behind my name like all these doctors and here, but I think I've got expert behind my name. I'm a mum and I'm going to continue the story for SMA. I was one of those parents. I had 2 children 18 years ago, Montana and Zali, and they both had type 1 SMA. They basically told me when they were born, your child has this disease, take her home, love her till she dies, there's nothing they can do. And that's devastating for any parent to hear. And through my journey, they were alive for 10 and 12 months. I promised and I vowed and declared when they did die that if I couldn't fix it for them, I would fix it for everyone else. And I've literally spent the last 18 years trying to do that for my children. And I'm very lucky that to say that I've been successful this year. So I want to take you through a few steps of what an advocate needs to do in the world of trying to get these beautiful medicines across the line. And the journey has to start somewhere. That's my journey. Why am I here? Why do I do this? Why are you here? What is your passion? My children were my passion, and I am very lucky that I get to leave a legacy on this world. And, you know, listening to everyone speak today, we all have a legacy to leave, and we all want to save lives. And, you know, I can honestly say in 10 years' time, people that don't even know about SMA right now, their lives will be saved because of my advocacy over the last 5 years. So you need to start early. If there is anyone in this room who needs to contact a patient organisation group or vice versa, you know, speak to a pharmaceutical company, make sure you're on the ball early. You cannot wait. You need to, this is a marathon, it's not a sprint. This will take a long time. Me getting or assisting to get or being part of the collaboration to get these treatments across the line was a 5-year process. So you'll have lots of wins, you'll have lots of losses as well. Stay in your lane and make sure that you've got your eyes on the prize, because there's a lot of people out there who don't want to see you succeed, and there's always a problem or there's something wrong. It teaches you about resilience. You know, you think you're sitting there, you're thinking, oh my God, why isn't this happening? You keep going. You've got to. There's an end prize at the end, and you've got to get there. The other thing I want to talk about is, you know, working with these people. How can you work with someone if you don't know them? You know, how can you work with someone if you don't like them? And how can you work with someone if you don't trust them? And I had to work with 3 separate organisations, 3 separate pharmaceutical companies, and I had to know, like, love and trust them to become their raving fan. And I was that person because each of them had a treatment that was beneficial for kids with spinal muscular atrophy. And it was without my input or the association's input that we were able to get these treatments across the line. And I think sometimes we undervalue patient organisation groups. Whether cold-face, we can get the real-world evidence. You know, doctors like Sandi and her team can get that clinical evidence, but we can actually see what's happening and survey families on the ground to make sure that it's actually doing what it's saying it's doing. Are you getting better? Are you improving? Are you declining? What's happening in your life? Is your mental health affected? There's all sorts of questions we can go and ask to make sure that what you're doing and what you're trying to get across the line is what they want. You know, those type of things. So lots of education, lots of communication, and coming back to your community to say thank you. You know, you need to tell them why you're doing it. This is for you as the patient. It didn't affect me at all. I can't benefit from any of these drugs, but I needed to make sure that I educated enough so that the families were able to understand why we had to survey them every year for the 5 years, what was happening in their world. So it's really important to give that communication back and make sure that you're telling them thank you. Why are we collecting this data? We're collecting this data to see who exists in our community. For example, one of the treatments for SMA didn't get passed because they didn't know how many people were in the community. So it's making sure that we're giving, you know, long-term in-depth data to make sure that treatments like gene therapies can, you know, be passed. Is it something that the community wants? And like Megan said before, you know, there's plenty of people in the community that know about it but don't understand it. We need to educate, we need to make sure that we create some baselines and make the community understand. See where that unmet need is and where the areas of concern are in the community. And this gives us actual real-world evidence and real-world data to make sure that we are aware of what's actually happening in the community. Is this transformational gene therapy doing what it's saying it's doing? Or are we just, you know, who cares? Do we not know what it's doing? So we need to make sure that everything that... And by the way, the community can collect this data quicker than what a researcher can. We don't have an ethics committee behind us that we need to, you know, get all the ticks and balances. We can be much more agile. For example, when gene therapy was coming through for the PBAC, it went from PBAC to MSAC back to PBAC. you know, that the PBAC were asking us questions. Is this really happening in the community? We could go out and we could survey the community, come back and give them the answer quite quickly, rather than waiting for an ethics approval through a hospital. So collaboration and making sure that you are getting that real world evidence and real world data is really important. It comes also back to credibility and validity of the patient perspective, because who's here? Who are we doing this for? We're doing this for the patient. So, yeah, and basically, like I said before, it informs the PBAC of what's happening within the community. So, not to sort of, you know, I'll put my results hat on now. This is what it can achieve, and we were in 5 years, and this is a very short time to get lots of different things across the line, but without you know, these bits and pieces, the SMA community wouldn't be changed like it is today. Historically, yes, we saw 1 baby a month die from this disease. That is now transformed. You know, you get a life of living, your child gets to grow up, you get memories with your child. In 5 years, we have got 3 treatments across the line, one of them being one of the first gene therapies for SMA in the entire world, and it's the highest cost drug ever at PBAC at $2.5 million per injection. So it's not easy, but it can be done. We also have been instrumental working on the National Newborn Screening Program behind the scenes, been working on that for 18 months, and I know that most of the states, all bar one, have definitely committed and are doing implementation programs next year, and I will not stop until I get the last state across the line, hopefully sometime soon. So, and we were also around when Mackenzie's mission you know, came along. We did a meeting in Parliament, ScoMo came that day, and things were changed forever. So it can be done. It's a lot of hard work, and the results speak for themselves. That little child that we saw before, me looking at that brings tears to my eyes, because that is not what I know SMA to be. I've seen my kids die from this disease, and that is truly amazing. So we can all play a part in this journey of gene therapy, and yeah, that's my story.
Thank you so much, Julie. So we've heard about a couple of different types of gene therapy, and we've heard about treatments that are going into patients. We're going to flip now and talk about things that are planned, and we're going to hear firstly from Dr. Tom Edwards. So Tom is a Melbourne-based eye surgeon who also leads a research team investigating gene therapy for inherited retinal diseases at the University of Melbourne and at the Centre for Eye Research Australia. Tom, can you tell us more about your work?
Thanks very much, Megan. And Julie, thanks for that inspirational talk on behalf of all the audience here. That was very moving and truly inspirational. Well, that's me. So I'm an ophthalmologist and a vitreoretinal surgeon, and also a clinician, sort of academic. I work up at the Centre for Eye Research Australia, which is based at the Royal Victorian Eye and Ear Hospital. And I'm very fortunate and privileged to work with a terrific team up there. Associate Professor Lauren Aiton was mentioned earlier by Megan, and she's sort of a co-lead of the group that I'm involved with. And we're really looking at primarily gene therapy for retinal diseases, and in particular inherited retinal diseases. Now, the eye is a really attractive target for gene therapy because it's a self-contained organ. The gene therapy can be delivered via standard surgical techniques directly to the retina without it needing to spread through the rest of the body, and is quarantined in the retina. The retina and the eye has a sort of immune privilege status in the body, so immune reactivity that it remains local to the eye, which is good. And the other great thing about the eye is you can see into the eye. I mean, we can look in and see the retina, the target organ, with our own eyes. And we've got a whole suite of high-tech imaging modalities to look at the anatomy of the retina and ways of assessing the function of the visual system to look for changes. Although for gene therapy there are still significant challenges that remain, which I'll mention towards the end. So this is a photo of us doing gene therapy now I don't remember if this was one of our clinical trial cases or it may have been the first case that we used Luxturna for, which was the first retinal gene therapy that was approved by the TGA following its FDA approval. And that's now a bench-to-bedside success story for retinal gene therapy. So it's a gene therapy specifically for one particular genetic cause of inherited retinal disease. Now there are over 200 up to 300 different causes, genetic causes of inherited retinal disease, but together they represent the most common cause of severe visual impairment in working-aged Australians. So whilst each gene individually can be rare, together they actually are quite, well, they're not rare, they're a relatively common cause, particularly in that working-aged Australian age group. So we've already had a good introduction to gene therapy, but at the risk of repeating that, essentially gene therapy is a catch-all umbrella term that encompasses different strategies designed to remedy or correct or block even a genetic change which is causing a problem for a retinal cell in this example and the research we're doing on retinal degeneration. So you can have a situation where you want to replace a gene that's not working as effectively as it should. So that's a typical scenario for an autosomal recessive disorder where you get essentially a dodgy copy from both parents, leaving you without the ability to produce a particular critical gene for retinal function. And so in that situation, you can just introduce a working copy of that gene. And that's typically done with use of a vector, a viral vector. And the one that's been used in a very large number of patients in clinical trials is an adeno-associated virus, particularly of serotype 2 or AAV2. And that's the one that's used for Luxturna. And so essentially what you're doing is you're hijacking the delivery mechanism of the virus, which we know when you catch a common cold is delivering viral DNA into your oral mucosa and your pharynx and giving you a sore throat. But in this situation, you're stripping out the majority of the viral DNA and putting the gene of interest that you want to deliver into retinal cells inside that virus. And you're using its predilection to target and effectively deliver a DNA payload into retinal cells. It needs to be done with a surgical approach, and you're generally using a needle as thin as a human hair to inject it underneath the retina, and then it delivers the genetic payload into those retinal cells. And as we heard earlier, theoretically that should be a once-off treatment for one eye, and then you can do the other eye. So that's gene augmentation. But there are other strategies. So you can edit your own genomic DNA using technology which you may have heard of, CRISPR-Cas9 technology, to sort of cut and paste, if you like, the mutation out of existence. And that can be done to correct a mutation and render the dysfunctional gene functional, or you can use a different strategy where you want to block a gene which has gone rogue and is causing problems for the cell. So you can block that gene's function and allow the cell to run off the other working copy. Because as you know, everyone has 2 copies of every gene, except the ones on the X chromosome and the Y chromosome. So that's an alternative strategy. And really you can then target downstream from DNA to RNA. So there are therapeutic antisense oligonucleotides that can target RNA and block or change RNA after the transcription phase. Or you can then, or again, you can block RNA from moving downstream and creating an aberrant protein. So that approach generally requires a regular intravitreal injection into the eye. And that's a strategy that's at late phase clinical trial at the moment. Another strategy for patients who, for example, have got quite advanced disease, because of course you need some functional retinal cells in order to deliver the genetic payload too. But if you've got the situation where someone's got very severe degeneration of the retina and the photoreceptor cells are all gone, there's another strategy called optogenetics, where you can deliver a genetic payload to cells in the inner retina whose job it is not normally to detect light, but you can infer light sensitivity properties to those cells, and they can take over the role of detecting light that has been lost due to the complete degeneration and absence of the photoreceptors. Now, of course, the vision's not as good, but for someone who's got next to no vision, every little bit counts. And so that's an alternative strategy. And it's not going to be a one-size-fits-all approach, because you've got people with different severities of disease. You've got people with, patients with disease caused by genes of different sizes, because not all genes are small enough to fit into an AAV, into its packaging capacity. And so it's going to be probably a tailored approach depending on the individual patient and the gene that's causing their inherited retinal disease. So one of the key challenges is measuring whether or not the disease has been affected by the gene therapy. Now, in this picture here, this nice immunofluorescence picture here, this is a mouse retina, and you can see in green they're the cells that we've transfected with or transduced rather, with a virus that's delivered a green fluorescent protein into those cells. So it's very easy to see which cells have been effectively treated, if you like, by that virus. But you can't stick a green fluorescent protein on the end of your gene of interest and have people walking around with glowing green retinas. And so that's a challenge for the community in terms of monitoring whether or not these treatments have been effective, or if they haven't been, why haven't they been? And because the disease has progressed relatively slowly, I mean that's a good thing for most patients. They're a slowly progressive disorder, but that means you need to follow these clinical trial participants in a clinical trial setting, or a patient in a clinical setting. You need to follow them for some time to determine whether or not the treatment has been successful. And that's a real challenge for the community, to work out good outcome measures that are effective at determining efficacy early on, so a clinical trial can move on, or indeed a patient can have some feedback as to whether or not the treatment's been successful. So one of the other key challenges is identifying the participants for clinical trials and the patients who might be eligible for these treatments. And a really important step, really a foundational endeavour, is for us to create a registry of all inherited retinal disease patients in Victoria and indeed Australia. And in fact, this is something that's going on all around the world, so that patients have the opportunity to register on a database, for example, and express their interest in getting information about upcoming clinical trials that they might like to be involved with, or indeed approved therapies as they come online. And so that's one of our big efforts at the Eye and Ear Hospital. We've just established in the last few years an ocular genetics clinic for patients with inherited retinal diseases, so they can come in and get a genetic diagnosis. And on the research front, we've got an ongoing registry and natural history study, which I co-lead with Associate Professor Lauren Ayton and a terrific team, as I mentioned. And the goal there is to create a registry of deep phenotyped and genotyped participants who we can then say to them, if there's an opportunity for a clinical trial coming along that is suitable for their particular genetic inherited retinal disease, then they're welcome to get involved. And lastly, I'll stretch out the sort of scope of gene therapy for retinal disorders beyond inherited retinal diseases, and we're fortunate to be conducting Australia's first gene therapy for late stage macular degeneration, which is a far more common disorder, affecting 1 in 30 of us over the age of 70 and 1 in 4 over the age of 90. So late stage AMD, the dry variety, not the wet variety, the dry variety of late AMD has no treatment. And whilst it's not caused by a single gene disorder like some of these other inherited retinal diseases I just mentioned, the gene therapy is designed to target the disease mechanism, the inflammatory mechanism underlying the disorder, to tilt it in favour of a more protective status rather than a more vulnerable status for disease progression. And so that's a clinical trial we're fortunate to be involved in, and we've got a number of terrific participants who've put their hand up for that, and that's an ongoing study. So it's not just inherited retinal diseases, it will broaden out to more common eye diseases as well. So that's all I have to say, and thanks very much for your time.
That's great, thanks Tom. It's a fantastic set of work that's being done at Sierra, and I think we'll have lots of opportunities to ask you more questions. But now we're going to hear from Anai. Dr. Anai Gonzalez Cordero is a researcher based at the Children's Medical Research Institute in Sydney and at the University of Sydney. Anai uses stem cells to develop new ways to study how eyes and ears develop in the laboratory and what goes wrong and how diseases manifest and how gene therapies could be used in the future. So over to you.
Okay, thank you everyone. It's a pleasure to be here. So I'm a basic scientist, a basic researcher with a passion about science, and but another passion that I have is to actually use the science and all the basic knowledge that we have and new discoveries that we do every day in the lab to translate that into new therapies and novel treatments. And hopefully I can convince you that stem cells and organoids that we developed from stem cells are actually very good models to test these new therapies. So just a brief introduction. So Ashley introduced blood stem cells, which are actually adult stem cells. We work with a different type of stem cells. We work with something that is called induced pluripotent stem cells. So these are one of my favorite cells in the body, not my favorites, but very special because they can divide in the dish, so in the lab, indefinitely. So we have a good bank of these cells. They can form every single cell of your body. Given the right instructions, they can form your eye, liver, heart, muscle, anything you can think of. And nowadays, we can actually get these cells from a blood sample. So we go back to blood, or maybe a very small piece of skin, and we can derive these cells using 4 different factors that were discovered this discovery won the Nobel Prize in 2012, and my God, Yamanaka, try to remember. So Yamanaka discovered the 4 crucial factors that are actually involved in stem cell development, and he discovered that these 4 factors are essential to transform the blood cells that we isolate in the lab, or the skin cells, into the pluripotent stem cells. So now they're called induced because they receive these 4 factors. And when you look down the microscope, they're there on that image at the top. You don't really distinguish them from other embryonic stem cells that in the past we used to obtain from human embryos. So what we do in the lab now, we grow these cells, we have a bank of these cells, and we can, based on our knowledge of development, and development, I'm talking about the knowledge that we have been obtaining from flies, fish, frogs, so all the beautiful developmental biology that was done years ago using these animal models, we can now apply it to our human cells. So one thing that I need to emphasize here, we can generate these cells from patients or from healthy individuals, and these cells have the same genetic background, right? So if there is a mutation on a gene of these patients, the stem cells will also have that mutation, right? So patients' own cells now generated into stem cells. Now we know what growth factors, what recipe to use to make these stem cells into the cells of our body because of our studies in developmental biology. And we can do 2 things. We can grow very simple, very simple cells on a monolayer in the dish, growing a bit messy, as you can see here, but we have a few light-sensing cells of the eye, so I also work in ophthalmology, and my job is to actually make retinal cells and the cells that actually sense light, the photoreceptor cells. So here you can see a bunch of the green rod photoreceptors there. But recently, and this was back in 2008, 2009, we were doing this type of cultures. Nowadays, we can actually make the stem cells into floating bodies. So basically just dissociate them, put them together into a small ball in the dish, and we give them the factors. The beauty is that because we formed that three-dimensional niche, these cells now spontaneously know how to form certain organs. And here I can show you a human organoid. So basically this is a mini eye that has been formed from the stem cells. Now the difference is that this mini organoid, this mini organ, has all the different cell types of the retina. They are laminated just like they should be at the back of your eye. They are functional when we grow them for a long time in culture. So the difference again is like in this case, in 2D, you cannot maintain them alive for too long. They don't like that environment because it's not the correct environment that they should be. But here we can keep these cells for over a year now, and they're functional. Now, what do we do with these cells? We test therapies. And one of them that we do in the lab is to test gene therapies. So here, Tom introduced the eye, and I'm just going to highlight the photoreceptor cells, so the light-sensing cells. Those are my favorite cells. Quite close to stem cells, but my favorite ones, because my job is to treat these cells. And if you lose these cells, or if you have a gene defect on these cells, you're going to go blind, pretty much. So what we do here, and I'm showing in green here, the cone cells, the ones that are responsible for color vision, central vision, so very essential for human vision. I'm showing them highlighted in green here. Again, this has been transduced with a viral vector, so it has received gene therapy. In this case, I used the viral vector to deliver the green protein because I wanted to separate these cells and transplant them. To do transplantation experiments that we also do, so cell therapy in this case. But what we can do is to basically test new therapies. So if you have a certain disease, for example, Usher's, that is one of the syndromes that we are trying to develop therapies in the lab, we can now design the viral vector, try and fit everything inside these vectors. Usher's is very challenging, so we actually have to use numerous AAVs or use the CRISPR-Cas and edit the DNA, as Tom mentioned before. And we can use the retinal organoids to test this therapy. And here is an example of the Usher treatments that we are doing. So basically, this is what the viral vector carries. So it carries the cutting machinery, which we call Cas9, the SA-Cas9, and a couple of guide RNA. So basically they're guides that are going to guide your Cas9 to the right place of the genome so it can go and cut and hopefully make, the cells recover. So here I'm just showing an image of the retinal organoid that was treated. So the beauty of the retinal organoid is that it's a human cell. So if you want to edit the genome, your guide RNAs that are guiding your cutting machinery are specific to the human genome. So you need to have a model where you can test these therapies. And I argue that the human organoids are the best models because they are human mini organs that closely resemble the native organ. And another disease that we have interest on, it's Stargardt's. So Stargardt's is a maculopathy, so defects in the macula. So the macula contain your cone photoreceptor cells. So it's an area rich in cones, and again is responsible for the central vision, color vision, and visual acuity. So very important for us. And Stargardt's is a disease that affects those cells in that area. Again, it's a large gene, so it's a challenging one. We can't just deliver with normal AAV and the replacement that Tom explained before. What we need to do in this case is to use two AAVs. So we add two different viral vectors to the mix, to the cells, growing in vitro, and we hope that they come together inside the cell and rescue the disease. So this is all happening in the lab still, with the hope that we can show efficacy in these human organoids, and then in a few years move on to clinical trials. And this is happening in parallel with a lot of other research going on, with other clinical trials happening. But again, here it's to emphasize the importance of stem cells and the organoids and how they can be beautiful models to test the therapy. And do I have anything else? I have, yeah. This is also not contained to the eye. We have a speciality in the lab, so once you generate organoids, you can pretty much play with your stem cells and try and generate other organs, and that's what we do. So we generate cortical organoids or neuronal brain organoids, and this one has actually been transduced again in green because we want to visualize the treatment with viral vector, and you can see how efficient it is in the neuronal cells. Here we have a heart. Is this going to play? So this is a video of a heart organoid, a cardiac organoid that beats. And here we have lung organoids, so airways. So just showing really the beauty of the models and how we now have thousands pretty much of human models that we can test therapies on. Thank you.
Thanks, Anai. So our final speaker is Dr. Peter Howling, and Peter is from the MCRI, from the Murdoch Children's Research Institute, and Peter has an interest and expertise in understanding muscle skeletal cells, and in particular muscular dystrophy. So Peter, over to you.
Great, thanks very much, and thanks for coming, and lucky last. It's always great going at the end after such brilliant talks, but we'll see how we go. So yeah, my name's Peter Howling. I'm a lab-based researcher at the Murdoch Children's Research Institute, and I study skeletal muscle. And one of the conditions or groups of conditions that we work on are the muscular dystrophies. And so muscular dystrophies can be caused by mutations in up to 60 different genes, over 600 different mutations. And while individually they are rare, as a whole they can affect up to 1 in 1,000 people worldwide. And one of the conditions that we work with is Duchenne muscular dystrophy. This is a muscle wasting and degenerative condition. And this is a photo here of one of the patients that comes through our clinic, Jack, and I'd like to thank him for allowing me to share part of his story today. So Jack was diagnosed with Duchenne muscular dystrophy at the age of 5. By 12, he had to use a wheelchair to move around, and by 15, he was confined to an electric wheelchair like what you see here in this picture. In 2017, he ended up in hospital, so some of the complications associated with muscular dystrophy not only affect your limb muscles, but also your heart and your breathing muscles. And during his time in hospital, he realised that really hospitals aren't set up for patients with a disability like his. And so since this time, he's been a really strong advocate for patients with muscular dystrophy to try and improve the quality of care that they get in the hospitals and how he can help patients who suffer from other conditions similar to Duchenne. And Jack's resolve in all of this is really what drives a bunch of what we do. And so I just sort of touch on a few aspects. And like what a lot of the other talks have spoken about before, it's really a team-based approach to what we do here. So we work really closely with philanthropic group Muscular Dystrophy Australia, and they provide key resources for patients with muscle diseases, educational resources and support. And we link in with them to be able to help give us focus and to work towards the treatments that we're trying to develop for patients with muscular dystrophy. We're also part of an Australian neuromuscular disease registry, and this is a really key aspect of what we do. It's a clinical registry that was established in 2020. It has over 600 patients included already, and that clinical information is really vital. But for a lab-based researcher, what we really need is patient material. And so for us, we're co-located at the Royal Children's Hospital, and we work with clinicians at the RCH to be able to contact patients who might be interested and involved in our studies. So we establish a muscle disease patient biobank, which includes blood samples from patients that come to the clinic. And there's two aspects that we look at. One is looking at the immune cells that are impacted in muscle disease. And what I want to talk to you today about is the models that we're developing using stem cells. And basically we're trying to create mini muscles in a dish to be able to represent what's happening in the patient. So we had a really excellent introduction around stem cells, so I won't go into too much detail, but basically we use blood from our patients and convert these to the induced pluripotent stem cells. And occasionally when we look down at our stem cells in the dish, they do smile back at us. This was a one-off account, but they occasionally interact with us, which is quite nice. But the benefit of these cells is that they can differentiate into many different cells of the body, and we're interested in trying to produce skeletal muscle. So skeletal muscle is important for motor function, and it forms, we use two different ways to model the muscle. One is a single layer two-dimensional structure here, so you can see the muscle fibers running parallel. Oh, can I point on that side? And the other is trying to develop more established 3D complex models, which is represented here. But the whole aim of this is to try to create patient-focused skeletal muscles. A lot of my work is focused on using mice as a model, and the other aspects that we can do is take biopsies directly from a patient. Now these have their pros and cons. Biopsies from kids is not something that is part of the standard clinical care anymore, so getting the material that we need to be able to study these conditions is really quite important. And stem cells really provide us that opportunity. So developing these models has been a big part of what we're trying to do. So this is an image of what a skeletal muscle looks like. And hopefully what you can see is one of the key aspects of muscle is that it has to contract. And so we stimulate these muscles in a dish using just a little electrical tickle to get them to contract. Hopefully you're able to see. So these are the fibers that run along the dish. This is our simple sort of 2D process. So if we can get these fibers to contract, we know we're on to a good thing, and then we can start to test some of the treatments that either improve the outcomes in the dish that we hope to translate to people. The other aspects that we're trying to do is to develop these 3D models. So these are the stem cells, muscle cells that we're growing around two little plastic poles. They sit up in the dish, and when we stimulate these to contract, the poles can move, and we can track the movement of these poles as a measure of force. So a lot of the treatments or conditions that we want to try to test, one of the things that we want to see is an output of increased force, and that's hard to do in a dish. But using this system, we're able to measure force. The other aspect of what we're doing here, so there you go. So as they beat, you can see the poles, the plastic poles moving. And this is, again, giving them a little electrical stimulation to encourage them to beat. The other aspect of these 3D cultures is they're more complex. Skeletal muscle is, not that it's a competition, but it's actually the biggest organ of our body, and you know, it's pretty important. It covers a lot of things. But it has a fairly complex architecture, not that we're competing with the eye at all, but the complications around the fibers interacting with each other and how they interact. These 3D models allow us to look at a bit more detail in what it would look like in the body. So ultimately what we've been trying to do is develop models that better represent our patients, with the idea that we translate any of these findings back to the patient. I just wanted to finish on this quote here. So this is Tugba. She's 22 years old. She was diagnosed at 6 months with congenital muscular dystrophy. And her statement really drives a lot of what we want to do, whereas she's saying research is important as it gives a sense of hope. It opens a world of opportunities. As the more research that is conducted, the more there is a chance to improve someone's quality of life. And so a lot of what we're trying to do is develop those tools and to be able to translate the findings that we have in the lab back to the patients in the best possible way. I'll finish there. Thank you.
So, over to you. Oh, that's a bit premature. I should go back. Just close your eyes for a moment while I just scroll back 20 slides. So what I want to do now is open the floor to you. So I'm sure these fantastic talks have raised some questions. If you want to raise your hand and keep your eyes off the screens for a moment. There we go. Yay. one of my colleagues, Helen and Caroline, will very happily give you the microphone. And to get things started, I'm going to ask the panel a question, because I thought it was really interesting hearing about some of the complexities and challenges. We deliberately chose to showcase a whole lot of different conditions today to really paint a spectrum of gene therapy, things that are already established, things that are still in development. And we've heard a lot about SMA, and we've heard a lot, and thanks very much both Julie and Sandi for giving us such a fabulous sort of insight into that condition and what it needed to get it there. But I want to go back a little bit and talk a bit about the science. So, you talked about sort of 5-year period to get sort of regulatory approval. But I wonder whether SMA was perhaps an easier gene to pack into the vector. Was there a technical limitation that made this a good target or a more effective treatment to get into patients?
That's a really good question, Megan. I think SMA is a good target condition because it has 95% of people have the missing SMN1. So 5% have a different genetics, but the majority are missing that SMN1. So if you can, and it is a relatively small gene compared to, say, a muscle gene like dystrophin, which is big. So it can be put into that AAV9 vector and delivered, and the motor neurons, it's transduced quickly into motor neurons. So on all those aspects, I think it is one of the exemplar diseases because it is so homogeneous in terms of the genetics that you can just replace the SMN1 and package it in AAV9 and deliver it.
Yeah, I can think Anna wants to say something.
Yes, about the AAV9. I think one of the major milestones that happened with SMA was that AAV9 was available at the time. And AAV9, so viral vectors have different capsids, so different flavors. And AAV9 is a good one because systemically reaches a lot of cells. So that was crucial for SMA treatment. In the eye, we started with AAV2, and we're now moving to other flavors that we know are a little bit more efficient.
And what's the difference between those numbers and the acronyms? What does that mean?
So the different proteins that there are in the capsids of the vectors. So that allows it to go into the cells.
And that determines kind of what cells will take them up? Is that effectively it?
Yes.
Ah, it's interesting.
They are quite ubiquitous, so they go into different cells, but there are preferences.
Yeah, and what's the challenges around using viral vectors? Are there things that needs to be taken into consideration, or if you've had a cold before, is that going to stop you?
Yeah,
using
so
the vector?
we are very cautious when we dose our children. So if they are unwell at baseline with cough, cold, kind of immune pro-inflammatory state anyway, we delay the onset of treatment. And we know that these gene therapies are systemic. They go to every cell in the body, but they do have side effects as well. So there are safety signals. A lot of inflammation in the liver is seen in our patient cohort. So we start dosing our children with steroids to calm that inflammation down one day before we give the gene therapy, and continue that for at least a month, sometimes longer. We see drops in platelets. So it's not a therapy that is without its side effects. There's a lot of inflammatory process going on. But in our unit at least, we've been able to manage those side effects.
And just remind me, how do you administer the vector to children? Is it intravenous?
Intravenous. So
Okay.
1 hour, the infusion takes around 1 hour. And I think the hardest part of the day for the parents is getting the cannula in, because we need 2 cannulas, because it's a $2.5 million drug. So we can't have the infusion stop halfway. So we get 2 cannulas in. Infusion is actually pretty straightforward. And then the monitoring starts. And the monitoring is really going to be lifelong for our children. Lots of bloods in the first couple of weeks, but then clinical reviews and annual liver ultrasounds as well.
And Tom, you were mentioning that an advantage of the eye is it's not systemic. So do you see any, because you're delivering it straight to the eye, do you see any kind of side effects outside the eye, or is it contained within?
No, it's entirely contained within the eye, but we have a similar approach to dampening down any local ocular inflammation. So depending on the protocol, we'll give sometimes we'll give an oral steroid, but almost always you'll give either an injection of a steroid around the eyeball at the end of the case, and then patients, as is actually the case after most eye surgery, will be on anti-inflammatory drops. So
and this inflammatory response, is that just because it's a response to a virus?
Yeah, you're trying to dampen down any provocation of the immune system in the eye.
Yeah. I was actually thinking when you were showing that beautiful green fluorescent retina, and you said that no one wants that. I reckon there's a lot of biohackers that will really love that. so I think that's something that we'll perhaps have to watch. But I wanted just to throw back to Ashley, because I know you were talking about CAR T, but we sort of didn't have much of a chance to talk about some of the work around haemophilia and thalassemia, and I just wondered whether you'd like to talk a little bit about the kind of ex vivo manipulation, so the manipulation of cells outside the body, because we've talked a lot about kind of using gene therapy to treat patients.
Yeah, so we brushed over, obviously, in a short talk about what's actually required to manipulate cells, to manipulate their genes, and has been mentioned by both our speakers about they need to be safe. And one of the things that we need to do is manufacture things because and give them safely under good manufacturing practices. And when we take a stem cell out of a patient and then have to manipulate the genes within it, we need to have all the processes from taking the stem cells out to making sure the stem cells are of good quality, to make sure that what you're actually doing to the stem cell is actually doing what you want it to do by manipulating the genes, by testing it, and then actually making sure that the product that you give back into the patient is what you expect it to be. So for all these types of cellular therapies, there are very stringent regulations from initial studies required even before you can actually try and do a study to place it into a patient. So, for example, there's, for CAR T therapies, only certain accredited laboratories are allowed to do it. And there's only one recently been opened in the Pacific region at MacCallum for CAR T cell therapy manufacture. Previously, that had all been controlled by the pharmaceutical companies, having very strict regulations about how those cellular therapies are made. And this has all come over decades of work, even manipulating stem cells to collect them, to reinfuse it into patients. We need to develop very stringent guidelines and regulatory processes so we can actually make it as safe as possible.
So in Australia, that's TGA? Yeah, Therapeutic Goods Administration.
But in addition to that, we have to report to a lot of different, for example, stem cell regulation for stem cells, different regulatory reporting to monitor our quality of what we're actually giving.
Is that around, so like an extension of usual bone marrow transplantation? Yeah. So sort of captured within that regulatory framework as well? Yeah,
there are lots of regulatory frameworks.
I believe we have a question from the floor.
Hello. Hi, panel. Thank you very much for your presentations today. Jennifer Hollins from the CDMO Cell Therapies at Peter Mac. I just wanted you to maybe take us through some of these therapies, ones that are marketed like CAR Ts, but also some of these gene therapies. They are very expensive therapies, but could you outline the case as to why these are worthwhile and how they could present new mainstream gold standard therapies that could potentially, move aside other types of therapies that may only provide incremental changes to patients?
Who'd like to go first?
I might start to answer that question. I think the elephant in the room is that these therapies are high cost, and at least in my realm of the world, they're rare neurogenetic diseases. How can you, when we've got such finite resourcing, how can you say this patient should get a $2.5 million drug? But what we have done is we've done cost economic modelling. So tying up a timely diagnosis, so that's through newborn screening, plus access to these disease-modifying therapies. And we have shown that they are actually cost-saving. They save lives. A lot of what we do in healthcare is low value. They are just incremental gains, but this is really transformative. You can see from that video, I don't need to say anything more. You save lives, you reduce hospital stay, you reduce the comorbidities that need a lot of support. We didn't go into the fact that these children need supplemental feeding, that they need ventilatory support for their lifetime, the allied health input. So we've found from our economic modelling that they are cost saving, and that's what's really pushed them through into the PBS system.
And if you compare it to a regular treatment, so we'll use SMA as the example. If you're giving them the first treatment that was around called Spinraza, it's $100,000 an injection. You use that over a lifetime of treating someone, that's millions and millions of dollars, whereas this is one injection once, it's $2.5 million once, rather than $10 million over 18 years. So the cost saving there is huge, and like Sandi said, it's transformational. This is, a treatment that kids don't have to go back into hospital for regularly to, their everyday living activities are outstanding. They're starting to walk, and historically, a parent living with a child with one of these diseases, you were at their bedside, trying to keep them alive more than you had them, doing everyday activities. So, that's the transformation that these treatments will bring to families in the future, most definitely.
I think you also have to think about not just individual cost, but what about the quality of life costs and societal costs. You are hopefully bring these children back into society and making their families productive again. So I think it's broader than just giving a medication worth millions to one child. It has such a ripple effect across society.
It also will give the children back their anonymity, that they won't be in a wheelchair. You won't be able to actually see in a classroom of children which one has SMA anymore. This is for our disease, I guess. So, yeah, the flow-on effect from that is also, really important to consider as well.
And I?
Yeah, just on the technical side, and I want to emphasize that this is one-off treatment because the vector goes in, delivers the healthy gene, and that's on, and hopefully on for a long time, for the lifelong of the patient. Manufacturing is very expensive, and there's not many places around the world that manufacture the viral vectors. So I think that we need to take that into consideration. That, of course, drives the prices up. There's queues to manufacture these vectors around the world. We are indeed now building a manufacturing facility in New South Wales, which will be great for Australia, gene therapy. So, yeah, there's quite a few things. It's gene specific. So the studies and all the technology that is developed, it's for a specific gene, one at a time. So as you can imagine, that also increases costs. So all of that taken into consideration just drives. And it's a new technology, and I think as the field evolves and as we manufacture more easily and it's more available, then hopefully the price will go down.
Is it similar in eye, Tom, that kind of cost?
Yeah, everyone's made great points. I don't really have anything much to add, actually, but I expect the price will come down, a bit like, all technology when it comes out, it's super expensive, but as the field becomes more competitive, the prices will come down, I hope, or I expect that will be the case.
So another question here from the floor.
Yeah, thank you, and thank you for your great presentations. Monica Ferry from the Genetic Support Network of Victoria, and our community, so the genetic undiagnosed and rare disease community, gene therapies and stem cell therapies do bring enormous hope. And Megan, you said at the start that 90% of people said they'd have them, but they didn't know what they were.
30%, under 30%.
Okay, still a high number. So my question to all of you, I guess, is because we need these therapies, and at the moment they're kind of under the radar a bit. Our patient communities are very, very aware of them and very pro, absolutely, let's create access. How do we do it faster? What can we do to work with you? How do we bring them into Australia, clinical trials, etc., etc. As the general community become more and more aware of the introduction of gene therapies, how do we bring them along in a positive way along this journey so that we don't find ourselves having arguments about genetics and eugenics and all kinds of things that are other elephants in the room, I guess? What can we do together to create a common message?
A big question.
What a big question. Monica, I think that hopefully that came out in my very short 7 minutes, that the community voice is the centre. We are so happy to work with our patient advocates. Without our patient advocates, we would not have got any of this through. So we understand that in rare diseases, genetic diseases, there is a big data gap. And I think just approach the researchers, the clinicians, and have a seat at the table, because that's what Julie won't take no for an answer for SMA. And we have worked collaboratively, and it is a community effort. So I think just reach out. And I think now the research space is paving the way for so much more collaboration. We know that we can't work in silos. So co-development is the name of the game. Co-development from the very inception of research.
Julie?
I also think education to those researchers, so going to see them, standing in front of them saying, hey, you've got the opportunity to save a life quicker if we all work together and collaborate. You know, how can we do that? And sit and have more discussion. I think we're not discussing it enough. You know, forums like this are fantastic, but we need to move to the next stage.
Ashley?
Yeah, I think things that we accept now as standard of care therapies, or having, for example, a bone marrow donor registry that I can ask for my bone marrow transplant coordinators to search if we have a suitable donor, and I can identify that donor within days. That actually came out from a patient advocate. So the International Bone Marrow Transplant Registry for Stem Cells, it was actually initiated from advocacy. So it's, as medical staff, we get stuck sometimes in the ways we do things. And what you're seeing here is the emergence of a new technology. And to bring people along actually does take a whole of community action and lobbying, yes, getting the evidence to show it actually works. And from that, and then moving that on to how do you actually scale it to get the right treatments to the right patients does take coordinated efforts. And no one group can probably do it by themselves. And it actually needs to have this unique alignment. So the fact that the approval was got within 5 years, to my ears, is actually quite incredible. Because if you take just like a pharmaceutical drug, the average time is about a decade to get from discovery to all the trials, to the regulation, and then into treatments. So that's an amazing time frame for a very novel therapy. And I think some goes back to also, if you think about gene therapy, people have been thinking for longer than 5 years. And perhaps we haven't touched on, we've talked about the type of vector virus that have been used, the adenovirus, but gene therapy back, let's say, from the early viruses, has been mentioned about the safety of some of them. They were actually not as safe. And for some of the ones that are used now, it's been grown out of experience to say that, and going through a lot of safety trials to say these are the safer ones to go through, and that's allowed the more rapid advancement and approvals. There are some of the earlier types of gene therapy where the viruses can insert bits of DNA in bits that you don't want. So next to something called oncogenes, and some of the early gene therapy
What would an
trials,
oncogene do, Ashley?
Yeah, it's a cancer-causing gene, so that's things that I'm interested in. So some of the early oncogenes were identified because the virus inserted just to stimulate the expression of these cancer-causing genes abnormally. And so down the track, some of the children with certain diseases, with immune diseases, started developing leukemias because the virus had gone into the cell's genome and stimulated the expression of a cancer-causing gene. So what we're doing here is actually learning through cold hard experience and clinical safety trials about what is the best approach now. And I think, with this approval and these types of viruses now, once we get past that safety mechanism and we can make those better assurances, then I think you can see the field increase and expand.
One more question from the floor. So I'm going to take the last question from the floor in a moment, but I just wanted to add to Monique's question that I think the other thing we have to be conscious of is that this won't be helpful for all. So I think that is another part of the message that we need to get out there, is the clarity about who and where and what it's going to do, to whom this can be used for and under what circumstances. So I think, I agree with you that we need to be working together, we need to be looking at these exemplars, but we also need to have it couched in where perhaps it won't be applied. And here I'm thinking about where there's more than one gene involved, for example. Last question from the middle.
I'll stand up. Hello, my name's Angela and I'm a medical scientist, but now I'm back in the classroom teaching high school students, and where Marilena and I are teaching VCE. So we now have an ethical overlay that is curriculum based within our courses that's coming up through from year 7 up to year 12. We are also teaching this data at year 11 and year 12 biology, and the ethical side of it is massive. Very massive, and I can only see you've touched on that, and you've presented 5 diseases, which is just fabulous to give us that diversity. But the question about funding, we have such limited funding in Australia for research. Who makes the ethical choice of who gets the money to do what? Is it the best presentation? Is it the best voice? And for Marilena and I, as educators in high school, as students who are going to be your future scientists, it's really important we get this message out to the community that, yes, one drug or one method is working now. This is just 5 years. Imagine what's going to happen in the next 5 years. And where is the ethics at a government, state government, national government, international level of whether we fix the faulty gene or whether we fix the different gene? Where's our overlay of policy and all of that over this? And the industry's gone so fast, what is going to happen if it gets out of hand in rogue laboratories and funding goes here or here or here? I know it's a big issue, but it's a real concern when I've got to teach my high school students about ethical choices. Yeah.
So I'd love your advocacy to government to get more funding. I'll get your number and talk to you afterwards. So for all types of research, and especially for clinical research, it's governed by human research ethics committees. And so that question about balance of benefit versus risk is always considered before we undertake any types of clinical trials in Australia. So that's an important consideration. I think also from the point of view of moving it forward in terms of scaling it to pharmaceuticals, there are a lot of steps and a lot of ethical considerations at each stage, from statistics to assessing benefit to identify whether a trial is continued or stopped. And also, as previously mentioned, all patients in a properly conducted trial are monitored principally for safety to make sure that they're actually volunteers in it, and it's conducted in a way that adhere to the principles of safe clinical practice and trial practice.
Such a big question to finish on, and I just wondered whether one of the panel would like...
Can I also add to that? the the funding will come from the federal government, so a lot of systemic change is done at Parliament, and you need to be talking to your local politician, your federal politician, to be able to educate them, and then they go on and educate Parliament, and that's exactly what happened in our sort of world here. They're the ones who make all the changes. You have to also understand that obviously there's a new change of, or there could be a change of government or a change of person in your local electorate, so it's a constant thing. Because the person might get ousted and you then start at zero again. So definitely education to those people, I would say, is really important to make them understand what generation is coming through, who they are, what they're doing, and how they need to be helped. Because if they don't know up here and you don't put your hand up and say, help us, they're just going to leave you out, is what I can add to that.
I think the other thing I was wanting the panellists to finish on is that question of where's the demarcation. So where and who decides on where this technology is being used? Can it be used for enhancement, for changing attributes, or should it be used only for life-saving and disease-ameliorating conditions and applications? So would anyone like to talk to that point?
I think it's enough of a... It's going to be enough of a challenge for us to deal with the diseases, the genetic diseases, before we get on to, some kind of lifestyle related, less health impactful treatments. So I think it'll all be driven by innovation from our scientists, and our next generation of scientists are in the classroom now, and it's great that they're being inspired by their teachers. Hopefully we'll see more of these treatments coming online. But I mean, one thing I just wanted to mention is it's important to manage patients' expectations here. The 10-year pipeline, for one gene, for one cause of inherited retinal disease, to use that example, and there's over 200 of them. It's going to be a slow process, and there'll be patients who it won't come, for their particular gene in their lifetime. So I think that's, not to end on a negative note, but just more of a realistic sort of point, I think, to make. I don't know, would you agree?
Yes, and I also want to emphasize the importance of very good preclinical research. For gene therapy, the first gene therapy in the eye, for example, which is our area, was done in 2000, so it's over 20 years of research for the very, very first one back then in the mouse eye, where we could see maybe a handful of cells that were transduced, not many, and just to see how the field evolved and the technology, but all based on very good preclinical data, and that's important. Education about what therapies you go and look for, stem cell research, it's really important that you do your research yourself and you know that the treatment that you're going to receive is based on good, solid data.
And I think the other thing we could probably just finish on is to say that this issue is taken very seriously by international scientists, by scientists around the world, and by governments. There are existing guidelines and frameworks around when and where treatment should be, gene therapy should be applied both to somatic cells, to the body cells, but also to embryos and or gametes. So this kind of ethical framework that is required is being developed as the technology is developing. It's not going to be, imposed, I think, once it gets there. So I think there's a lot of people already working this. There are a lot of international consortia who've already drafted and providing that guidance. So I'd like to talk to you more about that because it's right up my alley. And I couldn't agree more that we should be incorporating that kind of ethical and regulatory element and the patient and community engagement as we're dealing with these technical challenges. But whilst we didn't really focus on that today, I can assure you it's certainly happening. So I'm going to make a few concluding statements to firstly thank our fantastic panellists. I've certainly enjoyed tonight and learnt a lot, and I really thank them for sharing their wisdom and their passion. It's been wonderful. A recording of tonight will be available on the National Stem Cell Foundation of Australia's website, and also is being streamed on YouTube. And if you want to know more about this charity and the support that we provide Australian researchers, including Pete and Tom's work, and also many other activities that we're involved in, you can check out the website. And I've just been given permission to announce, you're the first ones to know, that both Anai and Ashley are the Metcalf Prize winners for this year. And this will be formally announced tomorrow, but the Foundation couldn't be more delighted to honour these outstanding researchers and to bring them into the NSCFA alumni. So we look forward to sharing more festivities with you this week. Finally, if you'd like to know more about the Australasian Society for Stem Cell Research, you can visit our website, learn about this network, and stay in touch with our community as the technology develops. We really want to hear from you and what you're interested in, and particularly we're interested in these misconceptions. We talked at the beginning about misunderstandings. Well, we're the scientists who know what's going on, and the doctors and ethicists, and we want to be part of that conversation. We want you to be part of that conversation, so join us. Finally, just make a comment that the Foundation and the Society are both committed to helping Australians make sense of the science, and so ultimately you can make a more informed choice about your healthcare and that of your family. So thank you very much for coming along tonight, and I think the panellists will be hanging around if anyone wants to come back for some more questions. Thanks so much. If you want one of our booklets on the way out from the Foundation, you're most welcome to pick one up from the desk.